Okay, so let's get started here. I posted another homework today, or I can't remember if it's a homework or I think it's a homework and not, or a journal and not an exam homework. Everything is graded. Um, if you are one of the Hamiltons, not Alexander, but I can't remember who. If you are one of the Hamiltons and you did not get a grade for the journal, let me come and see me afterwards because there's a problem with how it's getting turned in and processed by Blackboard. So if you're here, come and see me um, about that. Not everybody who should be coming to see me has come to see me, but I can tell you from the people who have come to see me so far, here, it's sort of what I'm guessing is going on in that coming up to this class, maybe you've had success with, waiting longer to study, getting away with doing somewhat less stuff. But the problem with this class is if you're not working on what we're talking about as we're talking about it in class and doing the problems at the back of the book, which every single person who's come to talk to me who's been having problems, first thing I ask them is, are you doing the problems at the back of the book? And the answer is no, and I say, well, you need to be, right? You need to practice what's going to happen on the test. It's fine to read the book. I mean, it's very good to read the book, take notes, watch over the lectures again, take notes on those. But when I give you the exam, it's not an exam over reviewing your notes or watching a lecture or reading the book. It's an exam over reading problem questions, figuring out what needs to be done, and solving those. So that's what you've got to practice. And that's why I try to encourage you. You can submit things individually if you want, but I try to get you to go into groups, and even if you are submitting the homeworks and journals and things by yourself, it would be good, especially if things are not going too well, to get into a group. Because the other thing that everybody who is coming in is saying, man, it's just not enough time, right? But this is the time for genetics, and you are in college and you need to make time for this. If you don't prioritize your classes and figure out how to do that, it's not going to be good. So being in a group is helpful with that, because when you're in a group and you set up a normal time to meet and then you don't show up, they can hold you accountable in a way that I can't. Where are you? Why are you not here? Help the person in your group to figure out that whatever they've got going on, unless it's a family emergency or something, like, hey, that's not a good enough reason to cancel and not show up to the group. Help each other to stay up with stuff. Hit the ISL, come see me. I'm happy to meet with a group of people. Like if you have a group, hey, we meet every Wednesday at 2 o'clock or something like that. I can't guarantee that I can hit any time, but Mondays and Wednesdays and some of Fridays, Mondays and Wednesdays almost any time is good. Fridays it all depends, because I have a whole bunch of meetings I go to and they change based on the week. But I can show up, or you guys can, at the end of your 40 minutes or whatever you're going to have as your review time and study time, I can come in and help you out at that, right? A lot of groups have had good luck going to the ISL, and you go into that, not the first room you go into, but the one over there on the left. You can sit at those tables and talk, and then if you have a problem, there's a tutor going to be there who can come over and help you just with that. So I want to encourage everybody to keep at it, keep going. Oh, shoot, I almost forgot to show you this. Also, it is super, can't emphasize it enough, important to read the directions on homework assignments and on exams and things like that. Whoops. It is cringeworthy for me when I'm sitting there trying to grade a homework or an exam, and I'm taking off massive amounts of points when you add it up over the whole class for something that you didn't do. Like on this last exam, or maybe it was one of the homeworks. Well, anyway, the last exam, when you were, I know this is for sure, when you were filling in the chromosomes, right? Half the points were for labeling the homologous chromosomes correctly, and there were a surprising number of people who didn't do that. Well, you just lost half the points. It's an F even though you got it right. And a lot of the people I could see, and I know that you could have understood it because you drew the chromosomes correctly. But you've got to do what you're supposed to do there. So let's look down. And I try to like put stuff in bold and underline it and all that. Look through those. This isn't in bold, but see what I have highlighted there? That was half the points. And there were a huge number of people who did not do that, who answered the question correctly, and I knew you could have done that, but you didn't do that. And so you lose half the points. I mean, you go down from 10 to 7.5. That's going from like an A+ to a C because I asked for something and you didn't do it. For example, this one here, right? Explain how variable penetrance and expressivity affects the nose wart phenotype. A number of people did a really good job explaining what variable penetrance and variable expressivity are, but did not mention the nose warts at all. And so you didn't answer the question. I can't help you. I can't give credit if you didn't do that. You know, stuff like that. All right, so that's enough of that. So that's enough of that. What I'm saying is, just please be very careful. There's plenty of time. There's definitely plenty of time with the homework. Reread the question. Make sure you're answering the question that I'm asking. Again, a big problem that students have is, and everybody has, because this is the way the human brain works, is that your brain, when you're reading a question, it's like just the facts, ma'am, but it's wrong. It draws what you recognize and you think you're looking for, and then just glosses over the rest. And that's how you're missing stuff. I mean, it's just a function of how the brain works. So you've got to try to train yourself to go more slowly. Reread the question when you're done. Get those points, right? I'm on your side with this. I'm concerned and I'm trying to help. You know, I want everybody to do as well as they possibly can. But I can't guess that you know how to answer a question if you don't do it. All right. So, last time we were talking about alleles that are in cis or in trans. How can I ask questions about this? There's pretty much only two ways that you can do this. Either I can give you this, the heterozygote, and tell you, that it's either in cis or in trans, right? Or just give you the picture here and not tell you that it's cis or trans. I can just give you like this drawn out, and then say that P and B are 20 map units apart. Figure out what are you going to get? And if you have this, then you're going to have 40% each of the chromosomes that have one dominant and one recessive allele, and 10% each for the chromosomes that either have two dominants or two recessives, right? So if I tell you this, it's still going to be the 40 and 40 for 80% parental or non-recombinant, and 10 and 10 are 20% recombinant, but these are going to change. Or the other way I can ask it is, I can tell you that you cross a green thorax brown puparium to a purple thorax black puparium and get these results, like, you know, the numbers and the words up here, but not that part that has the genes on it. What do the chromosomes in the heterozygote being tested look like? And you have to look here and say, okay, well, these two have to be the non-recombinants. And again, remember, the bottom chromosome is always the one from the tester, which is always homozygous recessive. So you can say, well, one of the chromosomes of the heterozygote has to be this one, and one has to be that one, so the dominant alleles are in cis and the recessive alleles are in cis, right? Or if I give you these results, you could figure out that the heterozygote had the dominants in trans and the recessives in trans. It's the only way, there's only two ways to do it. Make sense? All right. I also want to encourage, it seems like we're getting a little bit of a slowdown in people asking questions in class. I think I've already shown that it's going to be okay if you ask a question, and it's even okay and good if you're wrong, because when I ask a question and somebody answers it incorrectly, then figuring out why it's wrong helps you understand the way things should be. You know, correcting a misunderstanding fixes your thinking so that the next time we go into that topic, or you get it on a homework or on an exam, that you'll have everything set up. Okay. So, if you are given the recombination frequency, you can predict what you're going to get as far as the progeny from a cross. This is sort of a backwards and forwards thing, like I just talked about on the previous slide too. There's two ways I can ask these. I can either tell you what the recombination frequency or distance between the two genes is and ask what you would get from a cross, or I can give you the results of a cross and ask you to figure out the recombination frequency, which is the distance between the two genes. All right, so in this case, we're talking about cucumbers, and T and D. T is warty, big T is warty, little t is smooth, and D is dull, and big D is dull, and little d is glossy. So if you take a heterozygote and cross it to a homozygous recessive tester, and notice this is always the way it has to be. You have to be heterozygous for each gene here in the organism being tested, and homozygous here, because this homozygous tester is what's giving the, well, it's not showing right here, gives the bottom chromosome. Now, whether the dominant alleles are in cis here or in trans, it doesn't matter. You're heterozygous for both genes. It'll matter in the results, but yeah. All right, so in this case, when you do that cross, you're going to get 42% of each of the non-recombinant or parental gametes, and 8% each of the recombinant gametes. How do you know this? You know this by looking at the progeny. Remember the multiplication rule. The reason why we use a homozygous recessive tester is because the probability of getting a non-recombinant gamete that has both recessive alleles is 100% or 1.00. So the numbers that we get from these crosses are entirely dependent on the chromosome coming from the organism being tested. Right? So T and D recombine away from each other with a frequency of 0.16 or 16%. So 82% of the progeny are non-recombinant and 16% are recombinant. So you can figure out or predict the number of each that you get by knowing these frequencies. If you said, I did this cross and I planted 182 seeds, how many of those plants should produce smooth and glossy fruit? You can easily figure it out. The non-recombinant or parental is 84%, that's 42% for each. So 0.42 times whatever I said, 186 or whatever, is going to be the number that you get, right? So if I tell you the recombination frequency, you could figure everything out. The only trick here going this way is that if I tell you the recombination frequency is 16%, that means 16% are recombinant, 84% are non-recombinant, but you have to divide each one by 2. It's not 16% here, 16% here, and of course, you know, two of them with 84%, you can't have over 100%. So for any number of seeds that you plant, you can figure out exactly how many you should get, right? All right. So when you're talking about genetic mapping, using just recombination frequency to map the distance between genes, whoops, wrong button. 1% recombination is 1 map unit or 1 centimorgan, the little c, capital M because Thomas Hunt Morgan, right? I don't care if you say 1 map unit or 1 centimorgan, just pick one, but just know that both of those are there, and on standardized tests they're probably going to use this one because they're more gravitating toward the traditional way of saying things. So that means these genes are 16 map units apart. If I tell you that 2 genes are 16 map units apart, therefore you can go back and figure out all this stuff because the number of map units is the percent recombination. Change the percent recombination into a decimal, 0.16, divided by 2, that's 0.08 for each of these, and then the other ones have to be the total there. All right, so how would these percentages change? And so this is just for the picture. This stuff is all wrong right now. So what if I told you that these 2 genes were now not 16 map units, but that they were 22 map units apart? What does 22 map units mean? Is 22 is the percent recombination. So if there's 22% recombination, there's 22% recombinant progeny, and that must mean there's what fraction that is non-recombinant or parental? $78, right? Because 100 minus 80 is 100 minus 20 is 80. Subtract off another 2 is 78. So how many or what fraction of warty and glossy should you get? 0.1. So 10% this, 10% this, right? Or I'm sorry, 11, 22. And then whatever 78, is that 36? So 36% for each of these. Hopefully that's right. Because 35 and 35 is 70 and add another 2. So there you go. You can just figure it out. If you know how far apart a bunch of genes are on a chromosome with, this is called 2-point mapping because there's 2 genes involved, you can begin to build a genetic map. And if you have a lot of information about this, you can put it together like a puzzle. So let's look at this for a second. So maybe write this down because I'm going to go to the iPad here. You don't have to write down the names of all these fly mutations. Just write, like VG to CN is 9.5 if you don't already have it written down. Here, I'll just, ooh, there we go. There, hopefully that's legible. So if you know all these, you can put them together like a puzzle. What you can't do is know which one is all the way on the left and which one is all the way on the right. So we have to just guess it, right? When you put it together, you'll put these genes in order, you'll know the map distances between them, but you won't know which one's on the left and which one's on the right. So it could either be this way, the map, or just flip it around the other way. And the only way to know which it really is to get more mutants that go farther than that. So let's start putting the puzzle together. And hopefully I don't run out of space here. So I'll just throw that one in there. It helps to pick the smallest ones first, right? Now we're going to put another one with that. So let me mark this off as we've already used that one. So now you either have to go with another one that either has vestigial or curved in it, and the other one with vestigial is 9.5. The other one with curved is CU is 27. That's pretty far apart, the big numbers. So generally I try to go with the smaller numbers first, right? So here's what we can do. When you go VG to CN, so here's VG, but is Cinnabar eyes to the left or to the right of that? We can't tell. We do know it's 9.5 map units, so it could be over here, or it could be over here. Then you use the other genes to figure it out, right? So which other one has Cinnabar and curved on it? It's this one on the bottom here, right? See that guy? So if Cinnabar was to the left of vestigial, then the distance between Cinnabar and curved would be 9.5 plus 8.5, which would be 18. Ta-da! Right? And if Cinnabar is to the right of vestigial, the difference between the distance between Cinnabar and curved would only be 1.0, because this is vestigial to curved is 8.5, and then vestigial to Cinnabar is 9.5. We know those as facts. So we know that the one on top here, this mapping is correct because of the Cinnabar to curved 18. So then we can get rid of this. And mark this off and mark that off. And we'll go to the to the next one. So the next shortest one. So we have Cinnabar, which we've already seen, and BL is, I think, black body instead of gray body. And so the distance there is 9.0. So this is not going to be to scale. So Cinnabar to black, 9.0. Or Cinnabar to black, 9.0. That would, so the only way that we can figure it out now is the bottom one there, which is curved to black, is 27. So curved is over here. If black was to the left, you have 18 plus 9, which voila is 27, right? If that is not the case, then you would have to measure the other way. So black to vestigial is going to be 0.5 because we have 9.0 here and 9.5. Subtract that off. And so then you'd have 9.5 plus 8.5, which is 18, which is not right. So it's not magical. You just have to puzzle it out and put everything together. And so if you have a lot of 2-point mapping crosses, you can figure out how a lot of genes go together over a great genetic distance. So here we've got a total genetic distance up here at the top of 27 map units. And as we'll see coming up later on, you would never be able to do that accurately if you're just measuring from black all the way over to curved. Because of double crossovers, which we're going to talk about in just a second, the farther apart two genes are, the worse the 2-point genetic mapping based on recombination frequency becomes. So you want to do mapping of a whole bunch of gene pairs that are very close together and put them all together to make the bigger map like is shown here. Okay, I just alluded to the fact that mapping at a bigger distance is not good, and here is the reason. So we've talked about single crossovers already, or recombination events. If you have a recombination event that happens here, then one chromosome will have big A and little b, one will have little a and big B. If the big and little A and B phenotypes are easy to tell apart, then you can tell if recombination has occurred because you can tell if A and B are still together in the parental orientation or conformation, or if they've switched things around. But what if A and B are far enough apart, and they don't have to be that far apart for this to happen, that you get two recombination events in here? The first recombination, if it was by itself, would lead to big A and little b and little a and big B. But because there's a second recombination event, it puts everything back. And what you end up with is what's shown down here. There's an area that's been swapped between the A and the B genes. It's real, it's there, it happened, but you can't tell it happened because you can't see it. Because there's no gene in that area of red and blue that's been swapped that you can tell. So, map units are determined by the number of recombination events. The farther apart two genes are, the more recombination events because there's more space between them for a random recombination event to happen. But look at this. 2 recombination events happened here, and we're not counting either of them. So this is why genetic mapping gets screwed up as genes get farther apart, because you get double recombination events occurring and they're not counted. Not only are they not counted, but the second recombination event reduces by 1 the number of recombination events you count because it nulls out the first one. You can see this, however, if you're doing what's called a 3-point genetic cross. 2-point cross had 2 genes, 3-point cross has 3 genes. Now the 2 genes on the end, it's A and C because B is in the middle, the 2 genes on the end now have a 3rd gene in the middle. So you can detect single crossovers that happen between the left gene and the middle gene, like this, right? Or that happen between the middle gene and the right gene, or that happen between both. When I give you a problem and you're trying to figure out where the crosses occur, this is the way that's worked best for me to do this. Take your finger, put it on one of the chromosomes, and you're going to you're going to draw your finger from left to right across the chromosomes, move from one chromosome to the other where the recombination event happens, and draw out what you get. So here, big A, little b, little c. And the other one's going to be the opposite. Little a, big B, big C. Everybody see that? And right there. This one, little a, little b, and then big C. I know this seems like, 3rd grade-ish, but I do this myself. I run my finger across it even now when I'm drawing these things out. Big A, big B, little c. There it goes. For the double crossovers, it's especially important, I'm going to say to give it the finger, but that's, I know you want it, After you've worked a few of these, you might want to, but here, look. Big A, little b, big C. Big A, or little a, big B, little c, right here. So when you have 3 genes, you can detect a single crossover between the one on the left and the one in the middle, a single crossover between the one in the middle and the one in the right, and you can detect a double crossover as long as the first crossover is between the left and middle genes and the second one is between the middle and the right. If you get a double crossover between A and B, you will never detect it because you'll go big A and then down here and then back up, big B, big C. Little a, up here and then back down, little b, little c. Those are still undetectable. Got it? All right. So this is the way that genetic mapping used to be done. Mapping with a 3-point cross is much more accurate than mapping with a bunch of 2-point crosses put together because you can, to some degree, take into account the double crossovers. So nobody does this in fruit flies anymore because the genome has been sequenced, and we're going to talk about physical maps later on. So it's much better when you get to a physical map or the actual DNA sequence written out in A's, T's, G's, and C's than this genetic mapping. But it's still really important, and I keep this in my lessons because to understand how this mapping works teaches you how single and double crossovers work. So it's still doing a valuable service. All right, so in every one of these cases, we're going to be starting out with a fruit fly that has 3 genes on it. We're going to be crossing a female to be tested to a male homozygous recessive tester. Has to be that way because no recombination happens in Drosophila males. It's a weird thing, right? Because I said at the beginning that it's the recombination or the linking up of the DNA that holds the chromosomes together during meiotic prophase 1. And it's the covalent linkage of the DNA and then the cohesins holding the non-sister chromatids together. The male fruit fly doesn't have this, so there's some way that male fruit flies accomplish holding the chromosomes together, the homologous pairs, at metaphase of meiosis 1, but we don't know what it is yet, at least not that I know of. So here we go. We're going to have this setup. Now, you can do this cross just as easily if the fly being tested doesn't have all the wild type alleles in cis and all the recessive mutant alleles in cis. But to keep things simple, I'm going to always set it up this way. Just so you know, when you're working the problems in the book, one or two of them may have as a parental like ST plus ESS plus, right? A mixed up thing there. All right. So this is the way that the fruit flies people write it, with the plus is wild type and the lowercase is the mutant. So red eyes versus scarlet, gray body versus black body. E is actually ebony. That's what the E stands for. And spiny versus spineless. So these are spines on the legs. Then you get a thing like this. Or, whenever I give it to you and the book gives it to you, what you're going to get is these words and numbers on the right-hand side here only. And you're going to have to figure it out and put everything else in there. And they are not going to be in a really nice order like this. But what I would suggest, the way that I always solve these is that you put them together in groups. There's going to be 4 groups always. You can see, and here's the biggest group, here's the smallest group, and then there's 2 in the middle. Now, if the genes are very close to being the same distance apart, like if the middle gene is very close to being right in the middle, these numbers get pretty close and you can get mixed up as far as which is which. The book does not, and I will not ever give you a case like that. And if you got a case like that in real life, you would get a different middle gene because it would be too hard to figure out. So I would put them in the groups of 2, from the biggest at the top to the smallest at the bottom, so not like this. I would put these 2 together, then these 2, then these 2, and then these at the bottom. Okay? So you're going to go through this series of events that I'm giving you here. So I'm going to give you one of these in the homework. I guarantee this is going to be on the next exam. I don't know if it's going to be one of the big problems at the beginning and you work through all of them, but at the very least, I could put in the multiple choice part or parts of the big problems like little pieces parts of this. I give you some information and you figure out where stuff goes. So write out all the phenotypes and number of progeny produced, put them in groups of 2. It'll be easy to figure out which numbers go together. Put them from largest at the top to smallest at the bottom. Write out the genotypes of the original parents used to produce the triply heterozygous individual. That's just going to be essentially double up each of these chromosomes. Remember the bottom case in every one of these, see that it's the same? It always has to be there so you can figure out where the recombinations go by moving your finger between the genes. But remember that bottom one is just what comes from the homozygous recessive tester male. What you're concerned about is the chromosome that's on top in each of those cases, which is the one that comes from the original cross here, right? So there's usually two of them that have the most. Those are going to be the non-recombinants. That makes sense. Because the genes are linked, more will stay as the parental configuration than go to one of the recombinant configurations. So that's why I said put them at the top. Because the two with the most are going to be the parentals. And you can figure out which one was the original female parent just by looking at the one that's not all recessives. So this is what it's going to look like. Because everybody gets this from the other parent, and look, here's one of them, all three dominant. Here's the other one, all three recessive. So one of these chromosomes had to come from the female parent, and so did this one here, because they're on top. It will always be the one with the most progeny. And the reason is because the genes are linked. So remember what I said last time. If genes are unlinked, then it's just 50-50 because they're on different chromosomes. But if genes are linked, that means that they're together on the same chromosome, and even no matter how far apart they get, there will always be more that stay together than go apart until the genes get far enough apart. If there's two genes on either end of one of your chromosomes, they will look like they're on different chromosomes because they are so far apart that recombination between them is so likely that you can't tell whether they're on the same chromosome or on different ones. And that's why we do mapping of a bunch of genes. But here, you'll always find the non-recombinant parentals to have more than anything else. And it makes sense because they are on the same chromosome. They're more likely to stay together than they are to go apart. You can't have a recombination frequency that is higher than if they were on separate chromosomes. All right. Which ones are the double crossovers, the ones that have two crossovers? These are going to be the class that has the least. But you don't have to memorize that. Everything makes sense. So there's going to be some probability of getting a recombination between the left and the middle gene, right? Some probability of getting recombination between the middle and the right gene. What is the probability going to be of getting a recombination event between the left gene in the middle and the middle gene in the right? What rule do you use for that? We want to get this and that. When I say and, you think of what rule? The multiplication rule. If the recombination frequency between the left and the middle is 0.1, and the recombination frequency between the middle and the right is 0.15, then the probability of, in the same meiosis, getting a recombination between the left and the middle and the middle and the right is 0.1 times 0.5. That's going to be less than the frequency of either by itself. You can figure it out that way, or you can figure out, it just makes sense straight up thinking about it. The probability of getting a recombination between the left and the middle has to be more than the recombination of getting a left in the middle and the middle and the right, because to get both you have to get one of them first, and then it's a fraction of that. So the recombinant progeny, the double recombinant progeny, are the ones that have the least. So in this case it's the 5 and the 3. When you know these, you can figure out which gene lies in the middle. Remember the earlier slide, so I don't have to click back through all these. When you have a recombination between the left and the middle and the middle and the right, like A, B, and C, you'd get big A, little b, big C, and little a, big B, little c. It's only the gene in the middle that changes. So you can look here. Now, these aren't going to be in the right order, left, middle, and right. What's different between, so you can compare the parents here that are all wild type and look at these two. Well, this one has ST and E are plus and SS is different, right? Look at this one. ST and E are recessive and only SS is different. In both cases, you've figured out that SS is the one that's different, right? So therefore, that one has to be the one that's in the middle. Warning. You can look at every one of, except for this one, you can look at every one of these and get fooled. Look, here, E is different, right? ST and SS are plus and E is minus. For this one, it's ST that's different from those two. So in order to figure out which one's in the middle, you have to be looking at the two classes here of mutants that have the least number of progeny. All right? Or you're going to get fooled. Once you do that, you're going to rewrite the genes with that one in the middle. Actually, what I would strongly suggest is that you don't write down all this stuff until you get to that point, right? You can just write, well, you could just write these down and those numbers, and then you're going to put them in the right order later. So now we know that SS, or spineless, is going to be in the middle. So scarlet and ebony are on the left and right, or they could be on the right and left. We don't know. It could be either way. I'm going to ask you a favor for the homework and for on the exams. When you figure out which one's in the middle, which is spineless here, and then you want to put the other two genes on left and right, we're going to do it by alphabetical order with the one closest to A on the left and the one closest to B on the right. Do not put them all into alphabetical order, or you would have E, SS, well, actually it would work in this case, but in a lot of cases it doesn't. Put the one that's in the middle, and then pick the left and the right based on alphabetical order. All right, so now we've got them in the right direction here. So we already know that these two are the most, these two are at the least, and this is why I would say put them in this order to start with, because there's no sense in writing it out twice, is there? We already know that these two have no recombination. We already know that these two have two recombinations, and you can see here, look, this is in the middle. So if you start up at the top, I can't reach up there. From left to right, you can get scarlet, ST plus, SS nothing, and then E plus. And that's this guy right here. And the other one's going to be ST, SS plus, E. ST, SS plus, E. The only way you get those is to have a recombination between the left and the middle and between the middle and the right. Now we're going to look at these other two. One of them is going to have a recombination event between the left and the middle only, and the other one's going to have a recombination event between the middle and the right only. So the class with the most is double recombinants. The class with the least is, the class with the most is the parental or non-recombinant. The classes with the least is the double recombinants, and then these two in the middle are going to be the single recombinants that either have between the left and the middle or the middle and the right, but not both. So you can figure out where everything goes once they're in the right order by, again, drawing with your finger. And you can see here the red lines show where the crossovers must have taken place. So let's look up here. Between this one has ST plus and then SS and E. So ST plus, and then to get SS and E, you have to go down here and do that. So that's between the left and the middle. And you confirm it by looking at this one, ST and then SS plus E, ST, SS plus, E plus. The only way you can do that is to have one between the middle. And you could do the same thing with these two, and you'll get a single crossover between the middle and the right. Make sense enough? All right. I'm seeing funny looks. I'm not hearing any questions. Seriously good? Okay? All right. I mean, it's just following directions, but I want to make absolutely sure that you guys got this. All right. So now we're going to get started with our mapping and calculations. We're going to determine the recombination frequency. Again, I can't emphasize this enough. Recombination frequency is the number of recombinants over the total number. It is not the number of recombinants over the number of non-recombinants. So the recombinations between genes over the total progeny, multiply it by 100 to get the percent, which is the map units, and away we go. So for scarlet to spineless, which is the middle, the left to the middle, these two have a crossover between the left and the middle, so they are recombinant. 50 plus 52. But what's this? This is a tricky part. The spineless and scarlet are going to get counted in both recombinant progeny calculations. The reason for that is because they have a crossover between the left and the middle and between the middle and the right, so they're going to get counted twice. So 50 plus 52 plus 5 plus 3, there's 755 total. Multiply by 100, 14.6%. And from the middle to the right is these guys, because they're the ones that had a recombination event between the middle gene and the right gene. 43 plus 41, and again we have to count the double recombinants. It makes sense because they have one here, so you have to count it, and they have one here, you have to count it. Otherwise they would never be counted because there's not a double recombinant category. Alright, so that ends up being 12.2%. Now we're going to draw a genetic map showing the genes and the map units. You can add up the individual map units to get the total between them. So this is a lot more accurate for calculating the distance between scarlet and ebony than if you just used a scarlet and ebony cross directly. So now we're going to do another calculation. A double crossover requires a single crossover between here and here, scarlet and spineless, and between spineless and ebony, between there and there. If the first one happens over here and the second one happens over there, everything is fine. But what if one crossover happens here? Things could get a little weird. Or if one happens here. So crossovers are giant-sized events as far as, space that they take up. If a double crossover forms here, because of steric hindrance and size exclusion, it's going to inhibit a second crossover forming within a certain distance to the left or to the right. And over here doesn't matter because we wouldn't be able to measure that anyway. But over here, you could inhibit or interfere with the formation of a second double crossover. So we need to calculate the number of expected double crossovers based on these two numbers and then compare that to reality. So the expected number of double crossovers is calculated using the multiplication rule. As I said, you just multiply the probability of getting one between the left and the middle by the probability of getting a double crossover between the middle and the right. Right? 0.146 times 0.122. This is why the double crossovers are less frequent. 14.6% probability of getting a single here, 12.2 here, but only 1.78% probability of getting a crossover here and here. So how many would we expect to see? You take the probability and multiply it by 755, which is the total number, and you should get 13.4 double crossovers. Still with me? All right. Oh well, huh? Womp, 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 right? Sad trombones, because we only got 8. And the reason why we only got 8 instead of as many as expected, you could say chance can affect it a little bit higher, a little bit lower, but it's almost always going to be lower. And the reason is because of what I said. A crossover here might interfere with a second one over here, so it can't happen. And one here might interfere with a second one over there, so it can't happen. To figure out what's going on with this, we're going to calculate 2 more things. I know, it's the last 2 things, then we're done. We're going to calculate the coefficient of coincidence and interference. Coefficient is just the result of a division event, right? The coefficient of coincidence is the ratio of observed to expected double crossovers. And then we're going to use that number to calculate interference, which is the degree with which one crossover being in a certain area is going to inhibit nearby additional crossovers from forming. Yes? Oh, that's just right here. It's just you just add up the total number. And that's an important thing because, again, a lot of people are going to make a mistake, and that's why I'm telling you over and over again, you don't want to add up the non-recombinants and divide this by that number. You need to add up the everything, and that's where you get 755. That's the 755 here, here, and there. So here's the frequency with which you would expect one each fly that's born, I guess you would say. They're eggs. Here's how many you got total, so this is how many double crossovers you would expect for that number. All right. Okay, so the total observed double crossovers is 8. The total of expected is the 2 numbers here for individual probabilities multiplied by your total, 13.4, so 0.6. So what does this mean? And this is the important part because I always want you to explain what stuff means. That means that only 0.6 or 60% of the double crossovers that you should have seen actually show up. And interference is just 1 minus this, so interference just shows how many are missing. The interference is what is the fraction of double crossovers that you expected to get that you did not actually observe due to interference, and that's 0.4. So 40% of the expected double crossovers don't show up because when you get one over here, size-wise, it can interfere with the formation of a second one nearby. This seems like a lot, but it's just, you just go down through and follow the steps, right? And so I'm going to give you one of these on the homework. There are multiple of these in the book. Work through them. It's just the same thing every time, right? You figure out which are the non-recombinants, figure out which are the double recombinants, figure out which one's in the middle. That's the one that's the least compared to the most. Then calculate the recombination frequency from the left to the middle to the right, add them up to get both. Calculate the frequency of expected double crossovers. That's the product of the 2 frequencies multiplied by the total number as we just saw. And then calculate the coefficient of coincidence and the interference. It's a lot of stuff, But it's a lot of stuff, but it's a straightforward thing, and you can think your way through it. All right, so whether you're doing mapping with 2-point maps like we talked about earlier, or 3-point mapping we talked about now, the idea is you want to figure out what genes are on each chromosome. Now we just have the chromosome sequenced, but before you had to put it all together in what's called linkage groups. A linkage group is just a number of genes that you know are linked. That's all. This is chromosome 2 in fruit flies. The way they do their map units is they start at 0 at one end and then to whatever at the other end. So look at this. Halfway down the chromosome is 50 map units. Something is 50 map units apart, you should get a recombination frequency of 50%, right? So it doesn't really happen for reasons I'll talk about, but by the time you get down here, it's 100. You can't get 100% recombination. After you go over a certain distance apart, the genes up here at the top and down here at the bottom, because they are so far apart, if you tried to map net veins here and spec body down there at the bottom, it's not gonna look like they're linked at all. But we know they are. How do you do that? You make individual linkage groups, and there's a bunch of genes missing here, so there would be a lot more genes in there. Number 1, you can show that all these are linked together. Same with number 2, same with number 3, and same with number 4, but you can see that the individual linkage groups overlap. Not only do you know that curved wings is linked to all these, but you also know that it's linked to these down here. That joins group 4 to group 3. Does that make sense? Even though it would be very difficult to measure black body versus speck body because they're 50 map units apart, because you know that black body is connected to all these guys and lobis and curved wings are connected to all these genes at the bottom here, you know that it all goes together. So when you're individually doing these linkage groups, and you wouldn't be working on them all at the same time like this, then they can overlap into larger linkage groups, and eventually they connect it into the entire chromosome being one gigantic linkage group. So even though the genes at one end of the chromosome and the other end are way too far apart to show they're linked by mapping, by linking small groups, and the closer together the better, down the entire length of the chromosome, you're able to show that that they all do go together. All right, so we talked about double crossovers, and the one we talked about so far is this one at the top. So a 2-strand double crossover. 2-strand means the number of chromatids that are involved. Here it's this chromatid, the one that started out here is red, and the one that started out here is blue. If you have a 2-strand double crossover between 2 genes with nothing in the middle, then you can't detect any recombinants, because even though this middle part is all recombinant, there's nothing in there for you to look and see like gene-wise. 3-strand double crossovers are when you get a recombination between 2 chromosomes and then a second one between 2 other chromosomes. You can move your finger across and see what happens with these. In this case, you get 50% detectable recombinants. And so the reason for that is, so here you go with A, and then here, see how it goes underneath there? There's no second recombination event right here. So that's this one. You can tell that that's a recombinant chromosome. But this one, look, you have little a and it goes up to the red chromosome, but then it goes back down to this chromosome, and that's this one. So starts out blue, red in the middle, ends blue. This guy here starts out red and then goes to, or starts out blue and then goes red here at the end. So this chromatid here and this chromatid here each have only one recombination event, but this guy has one and then a second one. Because it has the second one, it's not, you know, it's these two are not and same thing here. These two are not detectable. So 50% detectable recombinants. You can detect this, and you can detect this one, but not this one, and not that one. And then if you have a 4-strand double crossover at the bottom, it's just like 2 individual single crossovers. The 2 middle ones have a crossover, you can detect those, and the 2 outside ones have a crossover, and you can detect those. So that's 100%. So the number of detectable recombinants averages 50% because the 3-strand is 50-50, the 2-strand is 0, and the 4-strand is 100. So if they're all equally likely to occur, then the total detectable recombinants is 50%. And that is why the farther apart you get between 2 genes, the more double crossovers you get here that you don't see. And so if you look at this, here is the actual map units if you were to do a bunch of little single crossover or single crossovers and map it. Here's the actual recombination frequency, so you can see it's linear, but man, it's probably linear up to like 10 map units. That's why I said you want to do a bunch of crosses of genes that are close together. But already by the time, so this is not even 20 to 20, by the time you get to 40, you're only like 27 map units. So they're 40 apart, but because there are so many double crossovers, you're only 40 map units. And look, it's sort of plateauing up here. So you're like 80 map units apart and you're still looking only like 40. So by the time you're that far away, it's only looking like it's about half the genetic distance. So the genetic mapping, as the genes get farther and farther apart, it lies to you. So you want to do a bunch of mapping with genes that are really close together. And actually, you want to get totally away from genetic mapping and just go to physical mapping, which we'll have to talk about next time. All right. See the lab people today. Everybody else, I will see you later. I'll post something, some homework for you to do or a journal for you to do.